The nucleus of a cell house is more than just DNA. It contains the essential components for genetic information, storage, and regulation. While DNA stores the information for all genes, molecules like histones and others apply epigenetic marks that regulate gene expression. We have roughly 20,000 protein-coding genes in our genome. But not every cell needs to make every protein. Cell specialization is key to life. Approximately 5,000 genes are considered housekeeping genes. These genes are active in virtually all cells. They include those required for essential processes like metabolism, transcription, or building the cytoskeleton. The remaining genes can be switched on or off depending on the cell type or environmental factors. A neuron, for example, has no business producing insulin. One crucial mechanism to switch genes off is DNA methylation. This process involves adding a methyl group to a cytosine base that is followed by a guanine. These places are called CpG sites. Because DNA is double-stranded, the complementary strand also has a CpG site, meaning both strands can be methylated. This is a powerful, heritable mark. When a cell divides, its DNA is copied. Initially, only the parental strand retains the old methylation mark. This partially marked DNA is called hemimethylated DNA. To ensure the daughter cell inherits the same silenced genes, this methylation pattern must be copied over. Let's see how this maintenance happens on a molecular level. The newly synthesized DNA strand lacks the methylation pattern. This is where UHRF1 comes in. UHRF1 is the key sensor that specifically recognizes and binds to that hemimethylated DNA. Once bound, UHRF1 acts as a beacon, recruiting the enzyme DNMT1, which stands for DNA methyltransferase 1. DNMT1 uses the molecule S-adenosyl L-methionine, or SAM, as its methyl donor. The methyl group on SAM sulfur atom is what DNMT1 transfers to the unmethylated cytosine on the new DNA strand. While DNMT1 can't reach the specific CpG site that is bound by UHRF1, it can methylate the multiple cytosines nearby. This works because CpG sites aren't distributed randomly. Instead, they're grouped into dense regions known as CpG islands, which are typically located near the promoters of genes. That's the ideal position to shut down a gene.